Title of my speech is Transforming Immunotherapy, a reverse pathway in B-lymphocytes for super-antibody technology and cure infectious diseases and cancer. Dear my colleagues, distinguished guests, and representatives of biotech companies of the world. I also, with light please, light is better. It is easy to say. Thank you. It is good. It is good. Light. It is good. It is good. That's good. No more light. This light is good. No, it is enough. My slide is easy to see. Let me also express my thanks to distinguished members of organizing committee of this international conference. Today, I will explain my new and right, I think that it is new and right, reverse pathway theory, which will change whole contemporary but wrong conception and clonal selection theory of molecular adaptive immunology. As you know, clonal selection theory, it is author of this theory until today we are using it. It is Sir Frank McFarlane Barnett, and he offered this theory, and until today we are using it. But actually, my findings also at the same time show that it is this theory, contemporary theory, clonal selection theory of Sir Frank McFarlane Barnett is totally wrong. Totally wrong. Not only one side is right, one side is wrong, but it is totally wrong. And after evidences I collected, existing evidences, also my theoretical what the conclusion was that it is totally wrong theory, and best theory I offered, it is reverse pathway theory, molecular immunology, adaptive immunology working totally different way. I called it reverse pathway mechanism. With this series, more nicely explain everything in adaptive immunology. And today, therefore, I will explain my new and right reverse pathway theory, which will change whole contemporary but wrong conception and clonal selection theory of molecular adaptive immunology. Background and discussion, several words about it. The contemporary clonal selection theory, which is explaining mechanism of adaptive immunology, alleging that the part of immunoglobulin gene in every naive B cell in bone marrow is undergoing so-called random rearrangements, and that way gaining unique affinity to one of the random antigen epitope even before meeting with its antigen epitope in reality. But it is totally wrong. Evidences and my theoretical calculation are showing that there are in all naive B cells are not going random rearrangement at all, but there is going some pattern rearrangement. also next evidences showing that clonal selection theory is wrong. For example, first, the clonal selection theory couldn't explain how and why is going so-called affinity maturation, and calling it somatic hypermutation phenomenon. But actually, if it is phenomenon, it means that this clonal selection theory is not theory, because theory have to answer all questions. And therefore, it shows that clonal selection theory is wrong. Actually, for my investigation, for my theoretical investigation, and evidence shows that in adaptive immunology, there is never happened somatic hypermutation phenomenon, as said. It has never happened. It is never existing. And therefore, it shows that clonal selection theory is wrong. Another proof, evidence is that clonal selection theory is wrong. It is that every infectious diseases and cancer have just definite number of antigen epitopes per diseases. Definite number. What's surface proteins, then go inside proteins, then go to nucleic acid antigen against nucleic acids. And it also shows that it is just impossible. Why impossible? Because if it is a random rearrangement, it means that random rearrangement is continuing. But actually, maybe patient catch some diseases, but maybe random rearrangement didn't prepare in enough immunoglobulin that matching with this antigen epitope. And also we have to remember that lifespan, half-lifespan of naive B cells is just only 1-2 months half-lifespan. It means that they are coming and dying. They are coming and dying after several months. And in this way, random rearrangement, it is impossible because it is coming, dying, coming, dying. It is not, there is not pool that existing every time, but it is also contradicting clonal selection theory. Third contradiction, clonal selection theory couldn't explain why all naive B cells have a membrane surfaces immunoglobulin M and immunoglobulin D. What are their real function? Actually, no immunoglobulin M and no immunoglobulin D never used as antibodies or immunoglobulins. They, for me, they use just only, I read a little more about it. And what's the real function of immunoglobulin M? They are not often to know an antigen epitope. No, they say that immunoglobulin have avidity but not affinity. But actually, it means that it is not using as a immunoglobulin of the B cell, don't using it as the immunoglobulin antigen epitope, often immunoglobulins. They use it just only for getting dendritic cells, one of the intraliminal vesicles. You know that dendritic cell is antigen-presenting cell, one of the, but everybody who read a little more about it knows that dendritic cell is the most powerful antigen-presenting cell than B cells. Also, a macrophage. Macrophage can induce production of some some of this antigen against antigen epitope. But thing is that I am saying that what my reverse pathway, I say theory, because it is really, because organisms do not have another way to explain all of this thing, just only reverse pathway. I will explain all of this reverse pathway, my theory, to your attention. Best very powerful antigen-presenting cell is dendritic cell. It is no B cells, no macrophage. Why? Dendritic cell constitutively produce antigen epitope on major compatibility 2. It is main constitutive function of dendritic cell. And dendritic cell is powerful, really, compare new synthesized naive B cells. And therefore, what combination is going in adaptive immunology? First combination, T cell and B cell coming to lymph nodes. And coming to lymph nodes, but they met with dendritic cells in lymph nodes. Because of dendritic cells came to lymph nodes from local infectious diseases, what's the site, or cancer site, they already prepared enough antigen epitope on major compatibility to. It is dendritic cells' main function to make antigen epitope and demonstrate it on major compatibility too. But this is T cells. About T cells, I say that there is also something going wrong. They say that T cells, immature T cells do not have specificity yet to antigen epitope. It is happening a little later. But actually, T cells never have specificity, against an antigen epitope. They just know their own antigen. It is endogen antigen. T cell have this ability. Why? Because T cell name is T, coming from thymus. It means that, but thymus have 3 layers, embryonic layers. Thymus is organized that way. It is ectodermal, endodermal, and mesodermal part. And therefore, T cells, which is learning all of their own embryonic whole spectrum of antigens, and they just know their own antigen and in thymus, but they do not specific, not to any foreign antigens. They do not have specificity. They know foreign antigen if it is no non-self antigen. They recognize all non-self antigen, microbial or virus or cancer, as a non-self self-antigen. What their function is, but not specifically now specific antigen, foreign antigen, never. Just all foreign antigen for T cells, it is non-self antigen. That's it. And he's fighting against that. How fighting? Just T cell, TH, actually, TH4 cell. What's the main cell who fighting against foreign antigens, and those are cancer cells. TH4 cells do not have do not have ability to go into dendritic cell and take one of the intraluminal vesicles. Intraluminal vesicles is vesicles which dendritic cell prepared very many, and they consist of major compatibility 2. On major compatibility 2, it is antigen epitope. What dendritic cell is doing as local antigen presenting cell, very powerful one. But TH cell do not have this ability to go and get one of the intraluminal vesicle from multivesicular body of dendritic cell. Dendritic cell have 2 layers of membrane, and he is hiding antigen epitope on major cell compatibility 2. You have to go in dendritic cell to take one of these. But B cells have this ability. And therefore, B cells surface receptor, all B cells have 2 surface receptors, all the same. There is, it is not working as an immunoglobulin. B cells surface membrane immunoglobulin-like things is immunoglobulin M and immunoglobulin D. And it means that if now sequencing method everywhere, we can sequence proteins, we also can sequence nucleic acids. If somebody will sequence and look to what's the immunoglobulin M protein, what kind of sequencing after sequencing structure is, and also immunoglobulin D, everybody will show that they are same. There is no differences, absolutely. Therefore, all naive B cells have just on cell surfaces same immunoglobulin M and immunoglobulin D, no other immunoglobulins. They have only 2 when it is producing in bone marrow. They do not know nothing. It is not specific immunoglobulin. It is just receptors, adhesive receptors, because all immunoglobulin has adhesive ability. It is that way. Or immunoglobulin superfamily, all members, adhesive molecules, also including this, not only immunoglobulin. And therefore, B cells having 2 additional adhesive receptor as the immunoglobulin M and immunoglobulin D can go interact with dendritic cell, can go through double membrane surfaces with adhesive molecule and get one of the intraluminal vesicles from dendritic cell, which dendritic cell as a local antigen presenting cell already had relation with microparticle and prepared this major compatibility to on this antigen epitope. It is already done by dendritic cell as a local antigen, powerful antigen presenting cell. And B cell getting it with the help to non-specific for antigen epitope. They just specific for membrane of dendritic cell. they can using adhesive ability go inside dendritic cell in membrane surface side, both membrane surfaces, and take from this membrane, double membrane surfaces, one intraluminal vesicles. Intraluminal vesicles, it is full of one type of, because there is many intraluminal vesicles inside of multivesicular body. It is actually endosome of dendritic cell, but called multivesicular body, because there is many intraluminal vesicles with antigen epitope on this major compatibility too, and only one type. Every protein from what's a foreign protein, there is going hydrolysis and partially enzymatic hydrolysis, and it is that way dendritic cell collected as on membrane surfaces or as intraluminal vesicles on major compatibility too. It is preparing that kind of things. It is dendritic cells' main function. And B cell have ability, naive B cell, I'm saying. All naive B cell have two receptors, immunoglobulin M more, another immunoglobulin D a little less. And with the help of this adhesive molecules, they work as adhesive molecules. They do not work as immunoglobulins, but we call them just immunoglobulins, M and immunoglobulins D. But mechanism is that adaptive immune system working that way. B cell, new B cell, coming in lymph nodes and spleen, follicles, and they meet with dendritic cell. And they actively going into interrelationship with dendritic cell and getting one of the intraluminal vesicles with full of antigen epitope on major compatibility to who Th4 cells need it. But Th4 cells do not have adhesive molecules. They cannot go to multivesicular body of dendritic cell. B cell taking it and then demonstrating it for also attracting. After taking this, one of the intraluminal vesicles from dendritic cell, naive B cell was very attractive to Th4 cells, because Th4 cells recognize major compatibility too on antigen epitope, but it has to be major compatibility on major compatibility too. What their function is, and T cell therefore attracting to naive B cells and have relationship because naive B cell taking intraluminal vesicles, naive B cells demonstrate included to its membrane surfaces, and it was very attractive to Th4 cells, because Th4 cells recognize antigen only on major compatibility too. And therefore, Th4 cells going not to dendritic cell, here around already, say going to B cells connection, because B cell already demonstrated very many antigen epitope on major compatibility too, which is Th4 cell needed. and actively engage. After that, first, what there is three main cells which we have to learn in the future for treating or curing cancer and infectious diseases. All B cell and T cell become active just only in lymph nodes or in follicle of spleen. What their function is, and they are non-specific until they do not engage with major compatibility 2 on on antigen on major compatibility 2 taken from dendritic cell. Dendritic cell then go naive B cell attracting, and naive B cell have adhesive molecules. It is not calling, we're calling immunoglobulins, but they work as an adhesive molecule, and they do not have specificity actually to any antigen epitope. And they going and taking into one of the interluminal vesicles, and then engaging with Th4 cell, because after taking one of the interluminal vesicles, they demonstrated, naive B cell demonstrated on membrane surfaces. And it is attracting Th4 cells, which is needed, this major compatibility antigen presenting. And therefore, there is going a relationship between dendritic cell first with naive B cells and taking with the help of adhesive molecules for one of the interluminal vesicles from naive B cells, from dendritic cells, and naive B cells demonstrated only naive B cells in membrane. Therefore, Th4 cell attracting not to dendritic cell because they do not have adhesive molecule, but they attracting to B cells, which in membrane surfaces already showing it to Th4 cells. And what's that kind of three cells begin creating adaptive immunity. Adaptive immunity do not creating as a random rearrangement as a clonal selection series. No, there is not a random rearrangement actually in a real part of immunoglobulin genes. There is not random rearrangement at all. There is pattern rearrangement just only. And then adaptive immune system begin with the interaction of three cells. First, most powerful dendritic cell. It is local antigen presenting cell, but most dendritic and have ability to prepare constitutively ability to prepare many antigen epitope or major compatibility to which Th4 cell needed, but Th4 cell cannot interact directly with dendritic cell because no other adhesive molecules as a naive B cell have. Okay, what three interaction begin showing how creating real and working adaptive immunology. What it's a second, I already told about it, naive B cell, showing what one of the interluminal vesicles in membrane surfaces. It is trogocytosis, so-called, and Th4 cell therefore become attract to naive B cell, not dendritic cell. And then after this attraction, then Th4 cells activate because already had many antigen epitope measures compatibility too, and it is activate Th4 cell. And Th4 cell is one of the very powerful antigen cytokine producing cell. It is very powerful one. It is Th4 cell, but activated by naive B cells. But then going cytokine production, many cytokine production. For example, some cytokine interleukin to increase 1,000 times. So just imagine how powerful the cytokine production begins from activated T cells, Th4 cells. And it is all this is, cytokines producing from activated Th4 cells activate naive B cells. And naive B cells, there is begin many synthesis of proteins and enzyme in naive B cells. Some of this even we do not know. It is main thing from my reverse phosphate theory was based because organisms do not have another mechanism. You cannot say that, no, you say no, right? Another maybe say another thing, but it is never happen because organism not randomly rearrangement using this for making antibodies, but organism giving particular to B cell exact genetic mechanism B cell give to immunoglobulin gene, exact mechanism, genetic mechanism from it is coming from the naive B cells. But how it has happened? How it has happened? It has happened because naive B cell prepare one enzyme on the cytokines of Th4 cells. It is unknown yet, but it is existing. I know that it is existing because no other way organism makes a specific antibody. And this naive B cell becoming centroblast after making this enzyme, after this enzyme using same transfer RNAs. Transfer RNAs, as you know, that making protein from nucleotides, as you know, it is translational from messenger RNA. But at the same time, transfer RNA gives possibility to make exact genetic code about the same make genetic code. What I am demonstrating this way. What it is in B cells. B cells producing one enzyme. It is RNA protein enzyme. Has to be. First time I think that it is reverse ribosome, but no. It is RNA protein enzyme, more simple, and when antigen polypeptide or measures the compatibility too, meeting this, there is many RNA transfer RNA in body. 500 gene working for transfer RNA. Transfer RNA gene, it is very many. Transfer RNA gene have active amino acids in one side. Protein synthesis using this as a protein synthesis. But in this case, this active amino acid interact with often amino acids of major antigen epitope. It is antigen epitope, what's it? Site, antigen epitope, it is also protein. Oops. And then active engaged, it interact with transfer RNA amino acid, active amino acid, and after making a good relationship, what the often relationship, but often relationship going in here, in B cell. After that, this enzyme up, it is unknown yet enzyme, RNA protein enzyme complex, using triplet anticodon of transfer RNA, and making short endosomal RNA. It is very specific. It is endogenic code of exact RNA for synthesis of often immunoglobulins. What this RNA organize triplets anticodon of transfer RNA. And then with the help of nuclear receptor, excuse me. Then with the help of nuclear receptor, this transfer RNA, what triplets of transfer action RNA, anticodon, with the nuclear acids, with nuclear receptors moving to VDJ part of immunoglobulin chain. It is only heavy chain have changed and becoming very specific antigen affecting producing immunoglobulin VDJ part of immunoglobulin, only going in heavy chain, not light chain. Light chain, it is additional chain. Actually, okay, let me finish. And light chain is additional, and antigen is big molecule, but antigen epitope small molecule interact with all the heavy chain. But last one I show. But all the heavy chain have specificity to antigen epitope, but not light chain, because it is too small molecule to compete with connect with antigen epitope. Heavy chain it is enough, and light chain connectedness non-specifically in another part of antigen epitope. But this connection, even non-covalent connection, even all connection, it is ionic, it is hydrophobic or van der Waals relationship, but not covalent relationship. They are coming, attracting each other, and then going away. It is, they have that kind of ability. But in body's pH, they become very tight. It is really a very interesting thing. It has changed central dogma of molecular biology. Why? Central dogma says that information, genetic information going from DNA to RNA to protein, but not from protein to protein, but not from protein back to RNA. But actually, immune system is working that way. They gave genetic information from polypeptide on major compatibility to exactly to DNA. And therefore, central dogma is changing. There is a possibility for some polypeptide protein giving back to DNA genetic information. That's it. What my reverse pathway is working that way. And my reverse pathway will make it possible to make very good often antibody against any microbes or cancer cells and cure patients with the help of exact synthesizing. It makes possible to synthesize in cell culture method. And therefore, it is very good method. Also, some patients have, for example, children also have, they are dying from mismatching what immune deficiency syndromes they have. And therefore, antibody treatment using cell culture, it is the best treatment, best cure, and there is have no side effect to patients. But that's it. Very unfortunately, I didn't have enough time, maybe. Thank you very much. I am really thanks to everyone.